Okay, the next speaker is Brbic. So she's talking about open world learning for biomedicine.
Thank you. Thank you for inviting me, and thank you for organizing this wonderful workshop. So my name is Maria, and I recently started my group at EPFL, actually 6 months ago. And today I will talk about open world learning for biomedicine. So with the advances of next generation sequencing technologies, we are nowadays witnessing an explosion of growth of biomedical data. Using single cell sequencing in particular, we can nowadays measure gene expressions in thousands or even millions of cells. And using very latest spatial technologies, we can even look how cells are spatially organized. So for the first time in history, we have really technologies to create a complete cellular makeup of the human body and understand what goes wrong on a cellular level in disease state. With this goal in mind, major consortia efforts has been created, such as Human Cell Atlas and Human Biomolecular Atlas program. And given these huge amounts of the genomic data that is being generated, naturally machine learning holds a promise for transforming biomedical research and really becoming the driving force of new discoveries in biology. However, the problem is that fundamental biology and natural science in general is very different than our prevalent machine learning paradigm. And there are two differences that I will talk about in this talk. So for the first difference is that in biology, we are often faced with a collection of small and different data sets that are generated under different experimental conditions, or in the machine learning language under different distributions. So biologists, for example, generate data sets across different tissues, different species, or individuals that suffer from different disease states. But in contrast, in our prevalent machine learning paradigm, we frequently assume that there is a massive data set that is generated from a single experimental condition or from a single distribution. Another challenge that is that in biology and in general natural sciences, we are really driven by new discoveries. So we want to be able to discover new and unknown phenomena that we don't know before. So for example, we want to be able to discover novel cell types or maybe kind of find new COVID variants. But in contrast, in our prevalent machine learning paradigm, what we typically do is that we predict existing and well-characterized phenomena. For example, we assign images to cats and dogs after we've seen examples of how they look like. But natural science and biology are not about really recognizing cats and dogs, but we want to be able to use machine learning to discover new phenomena, metaphorically saying discovering unicorns and phenomena that we have never seen before. So what biomedicine needs are kind of methods that can leverage these different and small data sets to generalize to novel experiments. This means kind of generalizing to new species, new diseases, new tissues, for example. And secondly, biomedicine also needs methods that can kind of discover new phenomena among already known and well-characterized phenomena. For example, discovering cancer cell states among normal cells. And this talk, I will kind of talk about how our research addresses some of these challenges. So in particular, we are interested in the problem of cell type characterization. And the reason why kind of single cell genomics brought such a revolution in biology is that, and the reason actually in general why we need this resolution of individual cells is that our cells are very heterogeneous. So they are very different from each other. And a fundamental computational problem is actually understanding these differences of individual cells that is known as kind of identification of cell types. And why is this important problem? So if we can characterize all our cells in the human body, this will help us to understand the basic biology and also what goes wrong on a cellular level in disease state. And this offers to have a profound impact on biology and medicine, ranging, for example, from disease diagnosis and prognosis, drug discovery, and drug efficacy and resistance. So in particular, what we are looking like at the problem is that we are given a matrix, which is cell time genes. And here we are measuring gene expressions in each individual cells. And this matrix is typically very high dimensional. So we can, using the latest technology, we can measure the full transcriptome, meaning kind of we can get almost like more than sometimes 20,000 or 30,000 dimensions in a gene space. And also and we can now do these measurements for hundreds of thousands or even millions of cells. And given this matrix, what we want to do is we want to kind of assign cells to different cell types, meaning we want to find meaningful groups of cells that kind of have similar gene expression profiles. And currently, there is a huge effort of the community really put to annotate these data sets. And the way typically this is done is that you have like people are generating these data sets in different experiments, and then each lab puts a lot of effort to annotate each individual data set separately. So in this case, how is this typically done is that you run some clustering algorithm, then people manually look whether these clusters are meaningful, whether they agree with a prior known knowledge of like marker genes that define particular cell types, and that manually annotate these cell types. And this, of course, is kind of very iterative because different clusters, we don't know how many clusters, so it's kind of very hard typically to do this. And our idea is kind of to reuse the data or kind of leverage a collection of previously annotated data sets to really help us to learn to generalize to a novel experiment. So what we want to do here is we want to design a learning algorithm that will take this set of previously annotated data sets and somehow leverage them to help us to learn better representation for a novel unannotated data set for which we don't have any annotations. And we want to be able to do this across very heterogeneous experiments, for example, across very different tissues. So why is this hard problem? Because each dataset may require solving entirely different learning tasks. For example, you imagine that in heart tissue, you may be seeing classes of fibroblasts and cardiomyocytes. Then in a pancreas tissue, maybe you've seen alpha and beta pancreatic cells. And now you want to be able to leverage somehow these datasets to help you to differentiate between different cell types in a brain. And maybe image classification analogy is like a bit more intuitive to people here, some people here. So what this means is kind of that I give you a set of previously annotated data set in which you see classes of jays, magpies, kingfishers, hummingbirds, gulls, and albatross. And now you want to be able to kind of leverage this information to help you to distinguish better and learn better representation that help you to differentiate between crows and nutcrackers. So the key idea in the MARS method that we develop is that we take a set of previously annotated data sets and then a novel unannotated data set. And what we do is we want to map all these data sets into joint low-dimensional embedding space in which we want to force cells to group according to their cell types or samples to group according to their classes. So in particular, what we do here is we take a high dimensional input dimensional vector, and then we learn a nonlinear embedding function F, which is a deep neural network. And also we learn a set of cell type prototypes or cell type landmarks in this embedding space. And these cell type landmarks are here annotated as these pentagons in different colors. And we learned two types of cell type landmarks for previously annotated data sets shown here in different colors, and cell type landmarks for novel unannotated data sets show in gray color because we don't know their identity. So what we want to do here is want to learn embedding function F such that cells from same cell types are embedded close together, while cells from different cell types are embedded far away. So to achieve this, we design an objective function that is iteratively optimized over landmarks and embedding parameters theta. And the objective function combines losses computed of the set of previously annotated data set and a loss computed on a novel unannotated data set. So in particular, for each annotated data set, what we want to do is we want to force samples to group according to their cell types. So we minimize the distance in the embedding space between the embedding of a data point and the ground truth cell type landmark. And our intuition here is that examples with the same classes should have similar embedding vector. And then at the same time, we want to force landmarks in the embedding space far away from each other. So we maximize the distance between the landmarks in the embedding space with the intuition that examples from different classes should have different embedding vectors. And then for each unannotated data set, what we do is we want force the samples to close to their closest cell type landmark. So what we do is we minimize the distance to the closest landmark in the embedding space. And at the same time, we force landmarks far away from each other to learn different embedding vectors for different classes. And the question we ask is, can this method MARS really generalize across different tissues? So what we do is we leave one tissue out, and then we use all other tissues as previously annotated data sets, and then we ask the algorithm to generalize to novel unseen tissues. So this means, for example, that we are using heart, lung, and pancreas tissues to help us to generalize to novel brain tissues that we have not seen before. And we apply this method to mouse aging cell atlas, which is the huge reference map of the all which characterizes mouse cell types and contains more than 100,000 cells across 23 different tissues. And we find that our method achieves 45% improvement compared to other methods that have been used for this task. And this is an example of the embedding space we get by MARS, where each cell type is really, each cells are naturally forming clusters, and they're embedded far away from different cell types, and same cell types are kind of forming nice same cells are cells belonging to the same cell types are forming nice clusters. Well, this is not a case, for example, in another method based on variational autoencoders, where you can see that different cell types are often separated, while same cell types are often separated, while different cell types are embedded close. So we then worked with the researchers at Stanford Medicine to really apply this method to discover neuronal types in the fly brain development. So we applied MARS to discover neuronal types at each developmental stage, and then also developed a methodology that allows us to trace this same neuronal type across development. And using our methodology, we were able to decode most of the neuronal types across different developmental stages. And we also worked to create the first single cell transcriptomic map of the entire fly. And very recently, we also published a very recent first characterization of the features of the aging fly, which was a huge effort, again, in collaboration with the researchers from Stanford Biology and Baylor College of Medicine. So in the follow-up work with what we were interested in is actually, can we also develop interpretable methods that, given, for example, novel tissue, we can also say, why are these cells assigned to particular cell type? For example, we wanted to answer question, why are these cells assigned to cardiomyocytes, and how are they different, for example, from fibroblasts? So to answer these questions, we looked at basically how do we humans generalize and reason. And we advocated that one of the core concepts on kind of how humans are really able to quickly adapt to new tasks lies in the concept-based reasoning. So, for example, if I ask you whether the left picture is a crow and the right picture is a raven, or the opposite, as humans, we are already equipped with some critical concepts that allows us to quickly reason. For example, we understand the concept of a beak, wing, and feather. So we can say, for example, the left bird is raven because ravens have thicker beaks than crows with more of a curve to the end. And we wanted to somehow use this concept-based reasoning, put this concept-based reasoning in a machine learning algorithm to be able to quickly adapt to a new domain, and also provide us human understandable interpretations. So the key idea here is that we learn along human interpretable concept dimensions. So given part-based annotations, what we do basically is when we learn the different embedding spaces for different parts of the image, or it could be also genomic data or textual data. But I here just visualize image for simplicity. And we call these different embedding functions concept learners. And then in different embedding spaces, we also learn different concept prototypes that represent, show how this prototypical concept of a particular class looks like. And we then compute compute different concept prototypes in each embedding space. And finally, I kind of combine these concept prototypes to get the final predictions. And this method is kind of extension of the meta learning method of prototypical networks, but it allows us basically to reason in terms of the in terms of the human understandable concepts, and also provide interpretations besides its predictions. So we apply this method to a few-shot learning problem in which we want to generalize to a new domain given only few labeled examples. And we show that this method is interpretable. For example, if I ask it why is a cell assigned to B cells, it can tell me that because the most important functional terms are B cell activation and B cell differentiation, but it also allows us to achieve state-of-the-art performance compared to other meta-learning algorithms, not only on the single cell data set, but also on the fine-grained image classification and topics generalizing, finding new topics in text documents. So far I talked about how can we use the kind of design methods that can leverage different and small data sets to really generalize to novel experiments. And in the next part of the talk, I will cover kind of how can we design methods that can discover novel phenomena among already known and existing phenomena. So in many real-world applications, and especially in biology and medicine, the space of novel and unknown is huge. And labeling data to train machine learning algorithms depends on having the complete knowledge of a given domain, which in biology and medicine is frequently not the case. For example, you can imagine that I know and have already characterized, there are some known cell types that I can already label, but also I want to be able to use machine learning algorithms to discover rare and novel cell types that have not been characterized before. So let me explain this in an example. So imagine I give you kind of data set coming from one donor, and in this donor, you will able to kind of find different cell types, kind of B cells, T cells, goblet cells, pneumocytes. So we were able to label according to existing knowledge these cells. And then you want to kind of train a classifier to distinguish between different cells in this cell types in this tissue. But then if I give you some kind of test data in the wild from a kind of another donor, and maybe this donor, for example, has a cancer, and this you want to apply this same classifier to a new donor, and you want to be able to kind of you can kind of characterize these existing cells that have been found in the healthy donor, but you also want to have the ability to really discover some novel cancer cell types and cancer cell states that have not been characterized before. So in particular, what we want to achieve is that you're given some label data in which maybe you find kind of fibroblasts and cardiomyocytes here, like this brown and red cells. And in your unlabeled data, besides these two types of cells, you also find some novel cells, for example, these green and purple ones that you have not been able to see before. So what you want to do is you want to say that these green cells are really novel cell type that you have not seen before, and these purple cells are also novel cell type that you have not seen before. And this is different than the outlier detection, for example, because you also want to be able to differentiate between different novel classes. So in particular, what we want to say is that we want to say that we want to be able to identify these existing classes, but also to discover these novel classes that we have not seen before. And we call this paradigm open world semi-supervised learning. So let me again make this analogy to the image classification. So imagine in your labeled data set, you've seen kind of jays and magpies. And then in your unlabeled data set, besides jays and magpies, there are also maybe nutcrackers and crows, like some new categories. So you want to be able to say that there is a novel bird species 1 and there is a novel bird species 2, but also to identify these existing classes of jays and magpies that you already characterized before. So to solve this problem, the key idea in our approach lies in really introducing this adaptive margin mechanism in the standard cross-entropy loss. So we combine the supervised objective, pairwise objective, and regularization. And particular regularization prevents assigning all examples to the same classes. But the key idea, as I said, lies in the supervised objective, in particular in the uncertainty-based adaptive margin mechanism that we introduce here. So we find that the core challenge in this setting is basically that discriminative features for seen classes are learned much faster than discriminative features for the novel classes. So the idea here is that we kind of control the learning speed of known classes compared to novel classes. So, and in this case, we use uncertainty to adapt this margin. So the intuition is basically that at the beginning of the training, uncertainty will be large. So we will use large margin, but close to the end of the training, and when uncertainty is small of novel examples, we will use kind of smaller margin. And then in the pairwise objective or unsupervised objective, this is based on kind of pseudo labeling idea, which has been shown previously to work well. So what we do is for the pairwise objective, we find the closest nearest neighbors in the embedding space, we generate pseudo labels, and then we use the binary cross entropy loss to train this part. And we apply this method also on single cell data sets, but also on the standard image classification benchmarks, and show that this method achieves a significant improvement over existing methods when they are extended to solve this task. And next, we've really applied this method to a problem in biology, in particular to spatially resolve single cell data. So spatial single cell technologies allows us also, besides to kind of measure expressions of genes, we can also look how cells are spatially organized in a space. So the question we ask is kind of, can we really annotate cancer donor tissue using kind of information from a healthy donor? So as a reference label dataset, this is the dataset that we actually trained on. We use the dataset from the healthy tonsil tissue. And here, these different colors denote different cell types. And as unlabeled dataset, we use the dataset coming from esophageal cancer, coming from the CODEX multiplex imaging technology, where you can see that the distribution of cell types is very different, but also you can see that there are some novel, for example, this major green cell type that has not been identified in this dataset. So to solve this problem and extend this idea of open world learning, semi-supervised learning to this problem, we use graph convolutional neural networks. So what we do is basically we assume that we are given a set of we are given an annotated reference spatial data set, and then we are given a target unannotated spatial data set. So what we do is kind of we construct the cell graph based on these distances of cells in the embedding space. And then we use the graph convolutional neural networks to learn embeddings that capture expressions of genes and proteins, but also the spatial arrangement. And finally, our goal is to really be able to discover novel cell types, but also to be able to characterize existing cell types. And these are the results when this method is really applied to the data set that I showed earlier. So these are the ground truth annotations, and these are the predictions we obtain using our method. And we show that our approach kind of almost perfectly agrees with the ground truth cell type annotations. And next, we collaborated with researchers at Stanford Medicine, in particular as a part of the Human Biomolecular Atlas program, whose ambitious goal is to create a 3D atlas of the human body. We really wanted to use this method to annotate cells in a spatial data set. So we applied this method in particular to data sets that I generated from colon and small bowel. And so far, Cellular has annotated more than 2.6 million cells coming from this consortia. We also validated whether these predictions are correct and find that they really agree well with what biologists would expect from this data set. And finally, in our very latest work, we asked the question kind of, can we really even go beyond? Can we kind of go beyond in a sense that can we transfer annotations across completely different species? And the additional challenge here is that in different species, there are different set of genes that are basically sequenced. And recently, there has been a huge effort to create these large scale single cell atlas data sets. For example, mouse cell atlas, lemur cell atlas, and human cell atlas has also recently been published. And this is an example of what happens, for example, if you want to integrate these methods using these datasets using existing approaches. So this is kind of dataset coming. Orange is zebrafish, and blue is frog. If you want to integrate these datasets using existing methods, you kind of have to match them genes using one-to-one orthologs matching. And when you plot them into joint embedding space, you cannot really find any kind of similarities between the cell types. You cannot transfer annotations, and you cannot understand how cell types are different or similar across completely different species. So to solve this problem, I will just briefly go over this. We propose a very method that we call SATURN. And the idea is this method is that we take a set of single cell RNA-seq data sets and also protein sequences. And the idea is that these protein sequences help us to overcome these differences in gene expression space. So what we do is we use protein language models and develop neural networks that basically learns these weights by that kind of encode the similarity of these protein embeddings in the space. So for example, what we want to do is that this macro gene here, that this internal space of the neural network that we call hidden space that we call macrogene space, the idea is that these macrogenes represent the groups of functionally related genes or proteins within the same species, but also across species. And in this way, we kind of want to map all different species data sets in the same embedding space. And we apply this method to kind of integrate multi-species cell atlas data sets, transfer annotations across different species, identify gene differences, and also find differentially expressed genes. And when we apply to the data set I showed you before, to zebrafish and frog, when we apply this method, this is the result we get. So here again, orange represents a zebrafish, blue represents frog. So you can see that now these cells are really integrated in this embedding space. And also here, the cells are colored based on the different cell types. So this means that we are really kind of matching also cell types correctly. And this allows us basically to transfer annotations, but also to study whether there are some like species-specific cell types or cell types are kind of conserved with the, across different species. And finally, most of this work was done while I was a postdoc at Stanford. So I would like to acknowledge my amazing collaborators I worked with, my postdoc advisor, Jure Leskovec, and amazing students who worked with me, Kaidi, Yana, Yusuf. Kaidi worked with me on COMET and Stellar, and Yana and Yusuf worked with me on SATURN. Thank you.
Let me ask a question first. To go back to the MARS method, so I want to see the embedding criterion. But the first term was basically the Fisher information, Fisher discriminant analysis. Right? So you want to embed
this one?
Yeah, samples in the same class, close, and other class is different. Then what about the unlabeled data? How did you handle it? I couldn't really understand why it generalized.
So for unlabeled data, because I mean this was a short talk, so I couldn't go into very much detail. So for unlabeled data, so we first pre-trained the network. So this is also important. So we pre-trained the network using kind of autoencoder based approach. And then what we do is like we run k-means clustering first in this embedding space to initialize the centroids to kind of get at least some meaningful initial data points. So for this unlabeled data, of course, we want to have some good starting point. So that's the idea. And then basically what we do is for unannotated data, we want to assign cells to kind of closest centroid or prototype in this embedding space. So you can imagine it is kind of like deep k-means clustering, basically. So we want to force cells close to this closest prototype. Of course, they will change as the algorithm the embeddings are updated, because in each iteration we update prototypes, we update embeddings, and also we want to force different prototypes far apart, so we maximize the distance between these prototypes.
So basically, pre-training is really important in that process.
Yes, but also because we compared, I'm not sure if I have the pictures here.
And also the number of clusters, how do you choose?
So maybe just let me, no, I don't have the picture, but so we also compared like what happens before, and then I will answer. So we had we compared what happens before pre-training and after pre-training. And before pre-training, like still some cell types, different cell types are sometimes grouped. So this fine tuning is like also really crucial also to find the good embeddings. But of course, if you just start immediately from, and you don't know how to initialize centroids, then your embeddings won't be meaningful. And for the number of clusters, so we have to, this is a parameter that we have to set, but it is, if we set it to some, like if we empirically find that if we set it to higher number, the method can lose the cluster. So at some point, if you like general intuition is like to set it a bit higher, so you can kind of lose some clusters, but of course you can never gain a cluster if you set it to the too small number.
Makes sense.
Yeah.
Thank you.
Yeah.
Okay, other questions? Yes.
Thank you for the great talk. So actually, we are also doing a close working with NIH to do the research on like the X-ray image, and then we try to find the subtype of the symptoms with sensitive to some drug and to some treatment. So for our research, for example, we find the symptom of the characteristics of symptom, whether like boundary is smooth or like what shape. It's very close to what you are doing, I guess. But in addition, because we also have to consider the relation between different organs. For example, pneumonia is not only pneumonia itself, it's also quite close to relevant to your heart. So I'm wondering, are you in your research, and I think you're only consider one like a single cell individually. Have you considered their relations in your current research?
You mean across tissues?
Yes. No. In the one tissue, for example, one cell may be connected, have some relation with another.
Yeah. They have definitely, because cell types are very hierarchically organized.
Yes.
So like T cells and B cells are, for example, both immune cells.
Yeah.
And then you can. The thing is that with like in this general annotation, like you can also go always on the finer grain level. For example, you can group T cells in the CD4 positive and CD8 positive T cells, right?
Yes.
So it's very hierarchical, and also there are also like a lot of discussion in the community are like about characterizing cell states, kind of a like current state in which cell is going, not only kind of cell type as its final state.
Yeah. So do you have any insight on how to also discover their relationship?
So we haven't done that so far, but I mean, for in biology, at least, like there are these kind of known, like there are these hierarchies. So maybe kind of using some prior knowledge in terms of kind of hierarchical structure and imputing this into the machine learning algorithm could somehow help to reveal, but this is, I think, still like a challenge in the community to really be able to find these fine-grained differences between different cells, because especially you are kind of in a very high-dimensional space. Sometimes you have to find one gene or few genes that are different between different cell types, which can be very challenging. And I think this is like there is still like new methods needed to kind of improve that.
Okay, thank you.
Thank you.
One more question.
Yeah, I'm interested in the concept learning in COMET. Yeah, so in the figure you show some parts are extracted. So these parts are automatically extracted, or it's given by human?
So that's a great question. So for example, depends on the data set. So for images, like for fine-grained image classification, we benchmark this on the CUP data set of birds. And for this part, we have the already part-based annotation, so we check how it works. But then we also tried with some algorithms that do this unsupervised landmark detection for, like, for example, flowers dataset, where we did not have these parts-based annotations. So it was also worked very well when you kind of somehow automatically get these concepts. And for gene data, for example, we already have these functional annotations of the genes coming from the gene ontology that you can use, and you want to be able to explain actually your predictions using meaningful concepts, which are kind of what people understand, like gene functions. For text data, for example, we were using WordNet to kind of find these similar words, basically.
Okay. Thank you very much.
You.
Okay, thanks, Maria.
Thank you.